ma'am.
Okay. How are you all?
Fine, ma'am.
What about others? What about others? Are you all there या फिर join करके चले गए? So I don't think I should be any bothered by who is attending the lecture, who is not attending the lecture, who is submitting the assignments and all. I shouldn't be bothered, right? Hmm? Can you all hear me?
Yes, ma'am.
Yes, ma'am.
Yeah, so should I be bothered by you guys not responding, you guys not submitting the assignments on time and all of that? I should not be bothered, no? I shouldn't care about those things, right? Because many of you all haven't submitted assignments. Should I start removing people who've not submitted their assignment?
No, ma'am.
तुम लोग ने क्या स्कोर किया है, that is secondary thing. Okay, that is secondary thing, because I mean it's okay, you all are still learning, so for me that is fine, but not attempting is a problem, and this has been brought to notice to Rujita and Pranav as well. Okay, so उनका तो सुनना पड़ेगा तुम लोगों को. So any doubts on the assignments should we discuss? Are you all interested to discuss the answers of assignment, or you all are least bothered?
Ma'am, we'll discuss.
What about others? Is Chaitanya your class representative? बाकी लोग के reactions का नहीं चल रहा है, chat box नहीं चल रहा है, audio नहीं चल रहा है. Yogesh, Dhanashri,
Yes, ma'am. Yes, ma'am.
very disappointing, you know. And if you all will, मतलब ऐसा response आएगा, then I wouldn't like, it won't motivate me to add on extra things. Like when I bring forth to you all things outside the textbook, extra things, is it relevant, or you all are like, ma'am, don't waste our time and don't waste your time?
No, ma'am, it's actually relevant. It's necessary for us.
But then this kind of response if I'm getting, then otherwise, let me straight off tell you all one thing. I've told this to earlier batches also, and I believe this is not our first or second lecture. हमने बहुत सारे lectures किए हैं, right, साथ में?
Yes, ma'am.
Yeah. I always tell the students, if you all are not interested to attend the lectures, don't attend. Some students हो गए मेरे lecture में, I'll be happy. I'll get one-on-one interaction with them, and it will be good for them as well. So if you all are not interested to attend, don't attend. Go and tell Rujita, go and tell the admins and all, okay, ma'am has allowed us to not attend the lecture because we are not interested. You all can watch recorded lecture. But if you all are here attending, then I want your 100%.
Yes, ma'am.
Yes, ma'am.
Advait, Amisha, Gaurav.
Yes, ma'am.
Yes, ma'am.
Yeah. So please
Yes,
don't
ma'am.
piss me off with this attitude of yours, okay? All right, then we'll discuss the assignment question. तुम लोग को कुछ doubt है उसमें? Because one, and did you all get the study material immediately?
Yes, ma'am.
Because, okay,
Yes, ma'am.
good, great, fine. Assignment, so directly, if you all realize, it's one question was from the slides which I've taught you all, yet some of them couldn't answer that properly. I don't know why. Another one was, वो भी मैंने तुम्हें बोला था, right, कि I will be asking you all this as an assignment, and those who've answered it correctly, those who've answered it on point, will realize it's from Remington, straightforward from Remington, right? तो ये जो मैं कुछ असाइनमेंट में देती हूँ ना तुम लोग को, record it, okay, मतलब record it in your notebooks and all, because they are also potential MCQ, okay, but I'm putting it as an assignment ताकि तुम वो section भी read करो, because if you see Remington में वो बहुत पीछे से needles के section में दिया है, we tend to skip that part, right? So my assignments will always be from the neglected sections of the topic, but which are important. So do it sincerely. It is going to benefit you all only. One more thing about the assignments, please don't upload it as an image. Please don't upload 2 files. Upload it as one PDF. Okay? I'll prefer image भी upload कर रहे हो, then one image में see to it दोनों answers आ जाए. Okay? Lengthy answers का जरूरत नहीं है. On point लिखो. And upload it as one image or one PDF. Don't upload 2 files. Okay? And do you all have an option of typing it?
No, ma'am.
Okay, there's no option for typing it. So जो भी तुम लोग upload कर रहे हो, upload as one file. All right? Let us now discuss. Is that clear, everyone?
Yes, ma'am.
Yes, ma'am.
Yes.
Okay. So I hope you all can see the screen here.
Yes, ma'am.
Okay, so this दिख रहा है ना? So yeah, this was the thing. Different conditions under which preservatives must not be used. I think किसी ने इसको misread करके कहाँ use करना है वो सब लिख दिया. Please pay attention to these words if they are used in the question are not used. So if it's a single dose which is exceeding 15 ml, मतलब large dose, that should connect you to large doses में और क्या आता है? Infusions. Okay? And single dose में नहीं चाहिए. And especially the ones, now some people have just written formulations for ophthalmic use. So उधर मैंने आधा मार्क कट किया है. It shouldn't be only ophthalmic, it should be ophthalmic and intracisternal or epidural and all of that. ठीक है? So that is the thing. And these are the correct answers for the needle size in inches and in gauge for the different routes of administration. Okay? So this is 1/4. Okay? This is not 1 or 4. 1/4. 0.25. Then this is 0.25 to 0.5. ठीक है? And these are the different gauge sizes. Fine. ये मैंने increasing order में इधर enlist किया है. So I will share these answers with you all as well. Fine. And what I'll do is I'll upload. नहीं, but तुम लोग को वो word file में नहीं है. Fine. I'll आज का जो slides which I'll be sharing you all ना, उसके starting में मैं या फिर end में assignment 1 के answers दे दूँ. Is that fine?
Yes, ma'am.
Fine.
Yes,
So आज का जो slides share करूँगी उसमें तुम लोग को ये दोनों assignment के answers मिल जाएंगे. So keep a note of that.
Ma'am.
हाँ.
Ma'am, could you please repeat? I lost due to my poor internet connectivity.
Nothing. I just told कि कहाँ पे preservatives use नहीं किए हैं. Understand the question. Okay. तो सिंगल डोज में जहाँ पे मोर देन 15 ml वॉल्यूम होता है वहां पे यूज नहीं किया जाता है. दैट विल लिंक यू टू लार्ज वॉल्यूम्स. ओके, लार्ज वॉल्यूम पैरेंटरल्स में वी डोंट यूज, which are infusions. Then you use it you do not use it in single dose, you use it in multi dose containers, and you do not use in ophthalmics or your CNS administrative pathways. Okay, so that's it. These are the things. So I'll share these with you all.
Okay.
Coming to now, we'll start today's thing, but I think before that we'll discuss MCQs for the last class. Do you all have your MCQ booklet?
Yes, ma'am.
Yes,
All
ma'am.
right. So open that. हम लास्ट लेक्चर का अभी सॉल्व करते हैं कि साथ में करना है बाद में? आई थिंक लेट्स डू इट नाउ. और हमें लायोफिलाइजेशन से चालू करना है, करेक्ट? So open your MCQ booklets, everyone. I want the correct answers to be posted on the chat box. Okay?
Okay, ma'am.
So you all can see this, right?
Yes, ma'am.
Yeah. So put your answers in the chat box.
Ma'am, screen is not visible.
स्क्रीन, हाँ, या जस्ट अ मिनट, वो बीच में डिस्कनेक्ट हो गया. अभी दिख रहा है, करेक्ट? तो सिलेक्ट ऑल द करेक्ट आंसर्स. चलो, मेक इट क्विक. So yes, the correct answer here is both. One. This is both infusions and implant. And what is the third thing? The third thing are injections, right?
Yes,
Yes,
ma'am.
ma'am.
These three are parenteral. Fine. Because here मैंने बोला है there can be more than one correct answer, इसीलिए वैसे है. Okay. Let's go to question 2. Select false. So read carefully and select the false. And please write the question number also when you all are typing the answer. 2 dash whatever. If there are multiple correct answers to one question, मैं इधर सामने से लिखा होगा. So in this, there is only one correct answer. So answer accordingly. Okay, fine. So most of you all have answered correctly. I think all of you. So select the false one. Presence of particulate matter rings an alarming bell. True. We don't want any particulate matter, right? So
Yes, ma'am.
ये वाला तो गलत है. Fine. Then quality of container and closure are less significant than API and excipients? No, they are equally significant, right? That's why we do inka extractable, leachables, compatibility tests and all of that, right? Then last one is formulation is made of simple ingredients, but process is complex. No, you also want the process to be as easy as possible, right? To help in keeping a facile process for sterilization and all. C option says pyrogenicity and isotonicity is not preferred. This is wrong, right? It should be preferred. So this is the false one. So the correct answer is C. Question 3. Samjha sabko? If you all have any doubts, stop me then and there. Which of these is advantageous with respect to manufacturing of parenteral? It's a very straightforward question actually. I hope you all are marking the correct answer and writing explanation in your booklets also. Are you all doing that?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Good. Yeah, so correct. I'm glad you all are answering it correctly. Even if it's a wrong answer, it's okay. But don't make it an irrational answer, okay? So yeah, the correct answer is D. Because because they are parenterals, because they have to be sterile, you need to have strict CGMP guidelines. So yes, this is right. Aseptic manufacturing process भी किया जाता है. कौन से products के लिए aseptic किया जाना जरूरी है? Like for which products aseptic processing is mandatory?
Ophthalmic.
Ophthalmic.
Why only ophthalmic?
No, parenterals also.
हाँ, but what type?
Ma'am,
हाँ, tell me.
ma'am, even suppositories are also should be of sterile properties.
We are not discussing that. Let's restrict ourselves to parenteral. Suppository comes under topical. Okay, we are not discussing about that. Aseptic manufacturing becomes important for products जो terminally sterilized नहीं हो सकते. ये मैंने last time भी बोला था. गांठ बन लो ये. जो products terminally sterilized नहीं हो सकते, okay, वो प्रोडक्ट्स को हम असेप्टिक करना पड़ेगा. मैन्युफैक्चरिंग मतलब पूरा मैन्युफैक्चरिंग असेप्टिक करना पड़ेगा. टर्मिनल स्टेरिलाइजेशन में क्या होता है? तुमने नॉर्मली कंपाउंडिंग किया, नॉर्मली कर दिया, एक लास्ट में टर्मिनल स्टेरिलाइजेशन मतलब लास्ट स्टेप पे तुम स्टेरिलाइज कर दो. ओके, बट देन देयर आर सम प्रोडक्ट्स जो एंड व्हिच आर द टेक्निक्स फॉर टर्मिनल स्टेरिलाइजेशन? टीएस, ऑटोक्लेव. ओके, दूसरा इज फिल्ट्रेशन थ्रू 0.22 माइक्रोन का मेम्ब्रेन फिल्टर. ओके, These are the two major ways of terminally sterilizing a product. But अगर जो thermolabile product है उसको तुम autoclave कर सकते हो?
No, ma'am.
नहीं. Then liposomes या कुछ microspheres या there are some products जो ऐसे suspension है उसको तुम sterile filter भी नहीं कर सकते हो. 0.22 micron माने कि 220 nanometers. What if your particle size is more than 220 nanometers? तो उनके लिए भी तुम नहीं कर सकते हो, right? So for those products which cannot be terminally sterilized, you have to do aseptic manufacturing process. मतलब step 1 से you need to maintain sterility. Clear everyone?
Yes,
ma'am.
ma'am.
Yes, ma'am.
There are many other clauses also for this, but at your level, these are the two important points which you all have to remember. Okay?
Ma'am,
And
can
then-
you repeat those points again? Those 2 points again?
What happened?
Ma'am, those
two-
2 points.
2 पॉइंट से ही टर्मिनली स्टेरिलाइज जो प्रोडक्ट्स नहीं हो सकते वहां पे तुम्हें असेप्टिक मैन्युफैक्चरिंग करना पड़ेगा. ओके, दैट इज द ओनली पॉइंट. Then sterilization of every component right from API, even this is required, correct? So all of these is the correct answer. Moving to question 4, which of these is true about infusion? मार्कर दिख रहा है ना तुम लोगों को?
Yes, ma'am.
Yes,
Okay.
ma'am.
Please make a note in your MCQ booklet. Which of these is true? This is right from the advantages slide. Thank you. Thank you for the correct answer. The correct answer is A. Drugs which are not stable in GIT, उनको तुम infusion या IV से दे सकते हो ना? Because they are not stable in GIT. तो इससे उसकी bioavailability भी बढ़ जाएगी. Infusions won't provide a localized effect. Injection at the site of effect will provide a localized effect. Example, dental injection in your gum. Infusion तो बकायदा हमेशा blood में ही जाता है. Infusions are IV, right? Infusions are always IV. They are always intravenous. Okay, so it will go in blood. It won't provide a localized effect. It will, on the contrary, provide what effect? What effect? Systemic. Thank you. It will provide a systemic effect. Faster onset of action than IV? No. IV will have a faster onset of action. IV injection. यहाँ पे मैं वो बात कर रही हूँ. Okay. IV one shot injection will have a faster effect compared to this. Okay. So the correct answer is A. Moving on to question 5. Select the wrong pair. This is also directly from the slides. तुम लोग स्लाइड्स खोल के बैठे हो क्या अभी?
No, ma'am.
Yeah, please don't do that. Yes, the correct answer is C. Shruti, what is the correct pair? Matlab, if this is wrong, what is the correct one? इंट्रा
Intra.
श्रुति है तू? Who is Shruti? Shruti suddenly went stealth mode. Huh? Ninja she is. One moment she is there, another moment disappeared. Shruti Patil. How about Rutuja? Thank you. It's intraperitoneal. These months there are many good movies coming up. So did you all watch Spider-Man or Flash?
No, ma'am.
No. You all are studying only the full time.
Yes, ma'am.
Come on, don't tell me you all are studying the full time. All the time nobody can study. Even I can't study all the time. Nobody went for Flash movie. किसी को Marvel या DC में interest नहीं है?
No, ma'am.
Oh my God, such a sad lot of students I'm seeing. Really? Nobody is into Avengers or Justice League or even the independent characters. Nobody. Oh my God. Mission Impossible?
I'm recently
No,
watched
ma'am.
Fast and Furious 1.
Oh my God. Fast and Furious 10 was bad. कुछ भी दिखाया है
Yes,
उसमें.
ma'am. Yes,
कुछ भी
ma'am.
दिखाया है उसने. मिशन इम्पॉसिबल भी किसी को नहीं पसंद. My deepest condolences to all of you for not watching good movies. ओपेनहाइमर के बारे में सुना है? क्रिस्टोफर नोलन फैन?
Yes, ma'am.
Thank God. Thank God. At least one person. Okay. Moving on. Question 6. Fun fact. ओपेनहाइमर किस चीज पे बेस्ड है?
I'm the Hiroshima Nagasaki.
Yes.
That
It's
effect that it had.
it's a story based on the scientist Oppenheimer, American nuclear scientist who worked on the atom bomb for America and which was used for Hiroshima and Nagasaki. You know, such weird type of questions come in NIPER, football and all that ka questions had come during my time. You all should watch some things other than your what do you say, mainstream academia. Anyways, question 6. Go and watch Spider-Man. It's animated movie. It's too good. You have that Shubman Gill who is dubbing for, I think, the Hindi thing. Shubman Gill, पता है सबको?
Yes, ma'am.
Yes, ma'am.
Yeah. First, yes, I heard from a girl.
Yes, ma'am.
Handsome cricketer, right?
Yes, ma'am.
Okay. So, what is wrong about subcutaneous injection? SC injection. It is given in the subcutaneous layer. Obviously, it is given in the subcutaneous layer. Administered in the hypodermis, right? Given at the angle of 25. So this is wrong. What angle is it given at?
45.
45.
Degree.
Good one. Good. All right. So moving on to the next question, number 7. Suspension cannot be given in dash. Read the options carefully. Okay. Shubham,
Yes, ma'am.
Okay. You have answered A. Why? See, here, if you all answer wrong, it's okay. I just want what are you all thinking. Okay. So, Shubham, why have you answered A? Shubham.
मैम
हाँ.
मैम ऑयल बेस्ड फॉर्मुलेशन रहता है सस्पेंशन में ऑयल ऑयल का ये दिया था ऑयल फेज रहता है इसीलिए वो कॉज करता है
Thrombophlebitis. Suspensions, हम emulsions की बात नहीं कर रहे. We are not talking about water in oil emulsions. We are talking about suspension. वो कोई भी parenteral में नहीं दिया जा सकता. Then what about the nanoparticles which you are making? Aren't they suspensions only? They are colloidal size range, but yeah, they are suspensions, no? Suspensions can be given through parenteral, but they cannot be given through, out of these options, they cannot be given through intracardiac. Intracardiac, IV, intra-arterial, sorry, arterial.
Intraperitoneal also, right?
Intraperitoneal depends.
Okay.
In certain animal studies, you give it intraperitoneal. Okay. IMSC mein, they are acceptable. So this is not true. You cannot generalize. Okay. Parenteral suspensions can be given parenterally, but they cannot be given through these routes. CNS wale mein bhi we don't prefer. All right. So these are the question 8. Shubham, did you understand? And those who've answered A, did you all understand why not A?
Yes, ma'am.
Okay, fine. Which of these is administered into the CSF in the subarachnoid space? ये बहुत direct question है. There's no logic to this. Okay, yeah. The correct answer is B, intrathecal. All right. Intrathecal is given in the subarachnoid space. Intraspinal,
Between the two vertebrae.
between the two vertebrae, in yours, it goes in the CSF only, right? Intracisternal,
Advent to cisterna magna
magna.
between atlas and axis vertebrae.
yeah, correct. Cisterna magna. Cisterna magna is the place, and the location of it is axis and atlas vertebrae के बीच में. Epidural,
The epidural part of the
हाँ.
body.
But then there are some things, right? Pia matter and this matter. Can anyone tell?
Between
Between
dura mater
pia-
and vertebrae.
Sorry?
Between dura mater and vertebrae.
Okay. And intrathecal में?
Ma'am, I said in epidural index.
हाँ. And intrathecal में? Pia matter में कौन सा रूट आता है?
Intrathecal.
Intrathecal.
Correct. Fine. Good. Yeah, this is not in your MCQ booklet, I believe. So you all can look at this and just answer it here. Where will the option A go? Where will option B go? Where will option C go? Okay.
What is the intravitreal injection?
1 minute, 1 minute, 1 minute, 1 minute. Sorry?
And what's the intravitreal injection?
In the vitreous cavity.
Okay.
Intracameral in the aqueous humor. Vitreal is in the vitreous humor.
Okay.
बोला था ना last time?
I didn't attend last lecture due to some issues. So
Okay. I hope you've got the notes and you've read it.
Yes, ma'am, right.
Good. All right then. Okay, first tell me subconjunctival में कितना है?
Less than 1 ml.
Good.
Less than
Less than 1 ml. Okay, so this is option A. Here. Intracameral में
0.1.
0.121.
0.121. Correct. And intravitreal में सबसे कम, less than 0. Okay. All right. So this is सबसे कम, this is moderate, and this is 1 ml max तक जा सकते हो. All right. So this is the thing. Again, the same thing. Preservatives are not used in single ml. Okay. Then intended into the eye and CSF, CNS related LVPs. So yeah, the correct answer is B. All right. We've not discussed lyophilization, but I think you all might be knowing the answer to this.
Freezing.
Freezing.
A.
Freezing.
A
yes.
option.
The principle of lyophilization is sublimation. It's not freezing. Freezing is the step. Okay, so we'll start our today's discussion from lyophilization. Freezing is a step. The principle for lyophilization is sublimation. The frozen water, that is ice, being directly converted into gaseous form at the triple point, so that what you obtain in the vial is water-free powder. All right. So this is the thing. Anyways, this is the last question, question 12. We are discussing lyophilization. So later, tell me the answer for this question, question 12. What is not true for infusion? Question 12. It's related to that preservative wala. Correct. Infusion contains antimicrobial is wrong. It do not. Okay, it doesn't contain. Infusions are large volume parenterals. Obviously, they are more than 100 ml. They're more than 500 ml, and they can be emulsion. They can be oil and water emulsion for sure. Okay. So with this, so we halt till question number 12. तुम लोग के booklet में I think it will be question 11, correct? Because उसमें वो ophthalmic वाला question नहीं था. And now we shall continue with the lecture discussion on parenteral, and we shall begin with lyophilization. So तुम लोग की study material में I believe lyophilization का slide है, right?
Ma'am, it is not uploaded yet.
Study material तुम लोग ने तो बोला it is uploaded.
Ma'am, it was lecture video.
Oh, lecture slides haven't been uploaded.
No, ma'am.
No, ma'am.
No, ma'am.
I have actually shared it, so you can maybe ask the admin. मैं भी remind करूँगी, but तुम लोग भी पूछ लो, ठीक है?
Yes, ma'am.
Because मैंने तो share कर दिया है. I will आज का content भी I will share immediately. So make sure you all like रात तक तुम्हें आ जाना चाहिए वो. If not, कल सुबह तो पूछ लो. Fine?
Yes, ma'am.
Okay.
Yes.
So can you all see the slide?
Yes,
Yes,
ma'am.
ma'am.
All right. So you all can see the lyophilization wala slide, right?
Yes, ma'am.
Okay. So lyophilization is a process which I said is to remove water, okay, from your parenteral product. So lyophilization को और क्या बोलते हैं?
Freeze
Lyophilization,
drying.
correct. उसको freeze drying भी बोलते हैं. The technical term is lyophilization. It's very important. All right. So what happens is that, tell me, a formulation in solution state is more stable or in a dry form is more stable?
Dry.
By
Dry
In
form.
dry form.
why dry form? Matlab we are talking about powder form. Why powder form is more stable?
Ma'am, the water or the solution ones are more prone to microbial contamination.
Even powder can be prone to contamination, but if it's an aqueous media, it is more favorable for microbial growth. Another reason, this is one reason. Another reason, ये तुमने माइक्रोबियल स्टेबिलिटी बोल दी. अभी लुक एट द केमिकल स्टेबिलिटी एंड आंसर. 3 types of stabilities are important. Okay. First is chemical stability. Actually, first is physical stability. Okay. The important one first is physical stability. Second is chemical stability. And third is microbial stability. So, Paneri ने answer किया, right? So, microbial stability का बोल दिया कि aqueous media is more favorable for microbial. What can you think about chemical stability now?
Yeah, powder form will be more stable because in solution form, it may affect due to the environmental condition and the storage condition of the solvent that we use. So that's maybe the reason.
You are going round the bush. I didn't get that right answer. Anyone? Look at point number 2 and answer. Point number 2 देखो screen पे, उसमें ही है answer. What is the answer?
Degradation by hydrolysis.
Exact. So we have studied post dispersions and colloids, right? Have we
Yes.
studied? Yeah,
Yes,
we've
ma'am.
studied. So उसमें मैंने क्या बोला था? Dispersions are of 3 types based on their size: molecular, colloidal, and coarse. And मैंने ये भी बोला था molecular is not really a dispersion because वो compound अपने native state में, okay, it's in its free form. And because whenever a compound is present in its molecular state, in its raw form, it is very easy to target it. It's the most vulnerable in its molecular state, in its raw form. Okay. Imagine a person being very naive, very native. Okay. In its, when they are at its raw form, when they are in the vulnerable form, कोई उनको कुछ भी आके बोल ले, they can easily feel bad. They will easily feel hurted. Right? So that is what is a compound being in its molecular form. In a molecular form, and when it is in its solution form, जब वो solubilized होता है, that means it's in its molecular form. Agree? Give me quick answers. Agree?
Yes, ma'am.
Yes,
Right?
ma'am.
When it's in a solubilized form, it's in a molecular form. So molecular form में it is very much susceptible to hydrolysis because environment water है, hydrolysis. Now you will say, ma'am, what if it's a non-aqueous preparation? What if it's not water? What if it's propylene glycol or ethanol? So उसमें भी there will be some amount of degradation because again it will be in its molecular form. Photolysis भी अगर होगा तो photolysis solution form में फटाफट होगा. Rate of photolysis will be more in solution form or in dry state? Solution form.
Solution
The
form.
reason being your compound is in its molecular state. जब भी तुम्हारा कंपाउंड मॉलिक्यूलर स्टेट में होता है, it is more susceptible to degradation, be it any form of degradation. All right, be it photolysis, be it hydrolysis, be it oxidation. All right, तो वो सब फटाफट हो जाता है. And physical stability can be a reflection of that. Change in color, agglomeration, all of those things.
Ma'am,
ठीक है?
also it will depend on the nature of
what?
in the solution state. Means whatever is the diluent that we use in the solution.
मतलब
So it may,
suppose a compound is soluble in water and तुमने उसमें solubilize किया है, then it will be more prone, right?
yeah.
If you for water soluble, अगर तुमने उसको इथेनॉल में डाला, पहली बात तो वो soluble ही नहीं होगा, suspended रहेगा.
Yes.
So the order of stability will be powder is relatively more stable compared to suspensions compared to the solution form. अभी ये solubilize तुम किस में भी करो, हाइड्रोफिलिक ड्रग को हाइड्रोफिलिक सॉल्वेंट में करो, लिपोफिलिक ड्रग को लिपोफिलिक सॉल्वेंट में करो, it is going to be solubilized, right, at the end of the day. And if it's solubilized, that means it's at the molecular level, and when it's at the molecular level, it is very much susceptible to degradation.
Okay.
All right, so to prevent this kind of loss, drug loss ही हो जाएगा ना तुम्हारा ड्रग. तुमने 20 mg पर 10 ml के वायल में ड्रग दिया है, डिग्रेडेशन से वो 20 का 10 mg हो जाएगा, तुम्हारी बायोअवेलेबिलिटी क्या हो गई, थेरेप्यूटिक इंडेक्स तो डिप हो जाएगा, करेक्ट, एक्टिविटी नहीं मिलेगी उसमें. सो टू प्रिवेंट दीज थिंग्स, हाउ कैन वी प्रिवेंट हाइड्रोलिसिस? पानी निकाल दो. और पानी कैसे निकालेंगे? बाय लायोफिलाइजिंग. ऑलराइट, अभी तुम बोलोगे मैम डायरेक्टली ड्रग एज अ पाउडर ही भर दो in the vial and then we can reconstitute it. Reconstitution का मतलब पता है? Not politically, scientifically. हाँ,
उसको वापस सॉल्यूशन फॉर्म में बना रहे हैं.
correct. Resolubilizing, reconstitution. Okay. Liposomes भी जब तुम बनाते हो, नैनो पार्टिकल्स भी जब तुम बनाते हो, they are suspended in state. उसको भी तुम लायोफिलाइज करके वो जो सराउंडिंग मीडिया है निकाल दो, तो तुम्हारा सस्पेंडेड वो सारा नैनो पार्टिकल्स या लाइपोसोम इन अ पाउडर स्टेट हो जाएगा. अभी उसमें तुम वापस से ड्यूरिंग, ओके, so transport, shelf life and all the time it's going to be in powder state only. Now while administering it to the patients, you reconstitute it with water or whatever the suitable media is. Again it will be in a suspended form then. All right, suppose if it was a powder, suppose it was a solution, उसको तुमने ड्राई किया and when you reconstitute it, उसका solution बन जाएगा. सस्पेंशन को ड्राई किया, रिकॉन्स्टिट्यूट किया तो सस्पेंशन बनेगा. सॉल्यूशन को ड्राई किया, रिकॉन्स्टिट्यूट किया, एट द टाइम ऑफ एडमिनिस्ट्रेशन इज गोइंग टू फॉर्म अ सस्पेंशन. ऑल राइट, so lyophilization is a technique of freeze drying using the principle of sublimation to obtain a stable product. All right, so they are used. Okay, so उसको तो lyophilized product का नाम won't be lyophilized. What will be written there is powder for injection or powder for infusion. And जब भी you have powder for injection or powder for infusion, उसको administration करने के time we will reconstitute it. Okay, we will reconstitute it and then we will inject it or infuse it to the patient. Okay, and it is given, it is done for the products which are not stable in solution. I explained you all the different things. Hydrolysis हो गया या oxidation, photolysis, anything. And in this process, a solution of the drug is prepared. Okay, so how do you make this? First, you prepare the sample. You prepare the sample, then you obviously sterilize it by whatever method. और इसको ही तुम असेप्टिक वे से प्रिपेयर करोगे। तो जो भी तुम्हारा स्टेरिलाइज पाउडर है उसको तुम एक लायोफिलाइजेशन वायल में फिल करोगे। इफ योर क्वेश्चन इज मैम, is the glass vial used for lyophilization and the normal glass vial different? Yes, they are different. These are more strong because they have to tolerate the temperature of -40, -50 डिग्री सेल्सियस। उतने पे हम फ्रीज करेंगे पानी को, तो उतना विथस्टैंड करना चाहिए। अदर ग्लास विल क्रैक, lyophilization vials won't crack. They are tubular type of vials. Okay, टाइप 1 ही होते हैं, बट देर आर एक ट्यूब्यूलर होता है एंड दूसरा आई फॉरगॉट। ओके, ट्यूब्यूलर एंड मोल्डेड, so these are tubular type of glass vials, lyophilization vials स्पेशली आते हैं। इसके स्टॉपर्स भी अलग होते हैं, इसके स्टॉपर्स में ना ऐसे स्लिट्स होते हैं, दिस विल बी द स्टॉपर। तो इसमें तुमने तुम्हारा सोल्यूशन भरा with use of lyoprotectants and cryoprotectants. Okay, then first step is freeze किया। freeze किया मतलब all the water in that will freeze and then you start drying it. So solid से gaseous state में जाने के process को क्या बोलते हैं? Sublimation. Okay, and this is done at the triple point of water. So find out what is triple point of water. And in this process, तुम्हारा जो freezing है, primary drying है, secondary drying है, okay? Primary drying and secondary drying करने के बाद you will get your final product, which will obviously be sterile. तो तुमने lyophilization machine में vial रख दिया जिसमें solution है एंड स्टॉपर भी लगा दिया, बट स्टॉपर को हम हाफ स्टॉपर करेंगे। हाफ स्टॉपर करेंगे ताकि जो भी फ्रीज होने के बाद जो गैस है वो ये स्लिट में से निकल जाएगा। दे आर स्लिटेड स्टॉपर्स। तो हाफ स्टॉपर किया तो वो स्लिट है, राइट? तो वो स्लिट में से गैस निकल जाएगा एंड आफ्टर सेकेंडरी ड्राइंग वो मशीन में ही फुल स्टॉपर कर दिया। तो जो लायोफिलाइजर, द मशीन विच जिसमें तुम लायोफिलाइज करते हो उसको लायोफिलाइजर बोलते हैं। तो जब तुम लायोफिलाइजर से प्रोडक्ट निकालोगे, व्हेन यू विल अनलोड इट, यू विल गेट अ ग्लास वायल विच इज स्टॉपर्ड एंड अंदर पाउडर हो जाएगा। भरा तुमने सोल्यूशन हाफ स्टॉपर करके, मिलेगा तुम्हें सील्ड, नॉट सील्ड, स्टॉपर्ड पाउडर फॉर्म एंड देन अल्यूमिनियम सील अलग से लगा दो। तो दिस इज व्हाट इज हैपनिंग नाउ। यहाँ पे जो अनीलिंग स्टेप है, अनीलिंग स्टेप इज एक्चुअली चेंजिंग द टेम्परेचर। मैं बस ऊपर से बोल देती हूँ, राइजिंग, रेजिंग एंड डिक्रीजिंग द टेम्परेचर। इट इज डन ड्यूरिंग फ्रीजिंग का स्टेप ओनली। अनीलिंग इज डन ड्यूरिंग फ्रीजिंग का स्टेप। इट इज चेंजिंग द टेम्परेचर, हाई लो करते हैं टू ऑब्टेन मोर स्टेबल प्रोडक्ट। This is done especially in cases of proteins for a proper freezing step. Proper freezing, complete freezing step में करते हैं। Okay? And in the process of lyophilization, your temperature and the vacuum or the pressure plays a very important role. So when you are developing a lyophilization recipe, it is called a recipe. Okay, when you are developing a lyophilization recipe, you need to optimize the temperature, the vacuum, the fill volume. कितना वॉल्यूम तुम फिल करोगे वायल में, कौन से साइज का वायल यूज करोगे, all of that is to be optimized. Okay, and the principle here is sublimation. Clear everyone?
Yes, ma'am.
Good. So this was about lyophilization. Now moving on to different components of parenteral preparation. So first is obviously the API, then is the vehicle which you are using, then are the other, what do you say, excipients, which are the preservatives, antioxidants, buffers, tonicity modifiers, surfactants, and also importantly your containers and closures. These also come under components. दूसरे डोजेज फॉर्म्स में पैकेजिंग मटेरियल एज अ कंपोनेंट एनलिस्ट नहीं किया जाता है. In parenterals, it becomes an important component. Why? I have explained you all in the earlier class, because it's in contact with your primary substance and because it has to go parenterally. That is why. ये सब sterile product होने के लिए सब important है. Understood? So we last class character जब हम properties पढ़ रहे थे, ideal attributes, उसमें हमने API के क्या प्रॉपर्टीज होने हैं वो डिस्कस की है, राइट? सो हियर नाउ we'll start with vehicles directly. So vehicles या solubilizers which you are using here, they are broadly divided into three. एक तो water ही है, तुम्हारा सबसे favorite is water, easily available. Then are water miscible solvents or vehicles, and then are non-aqueous solvents or vehicles. तो वाटर भी तुम्हारा WFI होता है, करेक्ट? वाटर फॉर इंजेक्शन इज द थिंग, एंड दैट you obtain water for injection either by the process of reverse osmosis or distillation. We discussed this last time. This because इससे तुम्हारा क्या निकलता है? Pyrogen. Because your pyrogens they are thermostable, तो सिर्फ हीटिंग से नहीं मरेगा, बट दे आर नॉन वॉलेटाइल एंड ऑब्वियसली फिल्ट्रेशन से भी नहीं जाएगा वो. दैट इज व्हाई वी डू डिस्टिलेशन. सो दैट इज अबाउट वाटर. सिंपल. वाटर मिसिबल सॉल्वेंट्स में यू हैव इथिलीन ग्लाइकोल, प्रोपिलीन ग्लाइकोल, पॉलीइथिलीन ग्लाइकोल. ओके. नॉन एक्विस में यू हैव योर ऑयल्स. ओके, यू हैव योर एस्टर्स. You have a range of solvents. Even NMP is one solvent, N-methyl pyrrolidone. Okay, these all are like widely used. Fine. Yeah, this was what I discussed about vehicles. So any doubt up till here? I think it's very clear.
Yes, ma'am. Clear.
Yes.
Yeah. So vehicles means if you're making, when you're trying to make a solution, I told you all, formulator will always try के उसका solution as simple as possible हो. That is drug plus पानी मिलके बन जाए and उसका stability अच्छा आए, sterility अच्छा आए. But the problem starts arising when solubility of drug in water doesn't come. Okay, so solubility of drug in WFI becomes a problem. So that is when the solution of using co-solvent like ethanol, glycerol, propylene glycol, PEG, glycerine, all that comes into role. So use of co-solvents or use of solubilizing agents like your surfactants, also your cyclodextrins, HP, beta, CD. Okay. All these, yeah, that is what cyclodextrin and many other techniques also will help you in a role. Comes called hello.
Yes, ma'am.
Yes, ma'am.
Yeah, so we've discussed that. You all can go through those videos to revise this. Let us just brush up some properties of your API. It has to be of parenteral grade. It has to be sterile and non-pyrogenic, right? It has to have low to nil microbial burden. मतलब नहीं होना चाहिए उसमें microbial. Chemical impurities नहीं होनी चाहिए. Endotoxin and microbial limits strict होने चाहिए. And it has to follow strict manufacturing protocol. So if I'm a formulator and if I'm purchasing API from some chemical company, then I look at the manufacturing protocol, I look at their COA, that is certificate of analysis, I look at their specifications. Specifications are nothing but the qualities. हम मोबाइल लेते हैं तो उसकी स्पेसिफिकेशंस ही तो देखते हैं, कैमरा का रेजोल्यूशन क्या है, ऑपरेटिंग सिस्टम क्या है, टच स्क्रीन कैसी है उसकी, ओके, इट शुड बी मोर पिक्सेलेटेड, राइट, DPI ज्यादा होना चाहिए, all of those things we look at the specification whenever you are buying anything. So even as when you are purchasing API or any excipients from any vendor, you look at the specifications कि उसकी assay value कितनी है. Assay value मतलब
प्यूरिटी
purity basically. मतलब उसका drug content should be like 100% that drug on, right? Impurity level low होना चाहिए, then heavy metal वगैरह low होना चाहिए, वो सारा ICH में उसके you will find the limits and all of that in ICH different guidelines. All right, so that is what is meant here, strict manufacturing protocols, because all these are checked. ठीक है. So now let us look at the different additives, different excipients. You know, my professor always tells me that excipients are like the masala of your formulation. They will enhance, okay? They will enhance and you use it very rationally. Kidhar kya use hua jata hai. And he also likes to call excipients as your hathiyar, your weapons. कहाँ पे कौन सा यूज करना है, कौन सा सल्यूबलाइजर कहाँ पे यूज करना है, कितने क्वांटिटी में, you know, that will change your formulation. It plays magic. All right, I'm very passionate about like making formulations, इसलिए I'm telling you all like this. But believe me, this was my experience. मेरा एक patented product है wherein the solubility of that was like 50 mg, but we used an excipient, okay, very commonly used. We use that excipient in a different way, and 50 mg का हमें 150 mg 200 mg आ रहा है solubility, almost 3 to 4 times आ रहा है, and that's a big deal. All right, so these are the role of excipient. This is the importance of excipient. All right. So even though if you'll go into marketing and all of that, na, you'll need to present your products in this way. So I hope मेरे lectures, if not scientifically, at least in other way, कभी काम आए तुम लोगों को. Anyways, so let us look at the additives based on their functionality. Okay, so this is the classification based on their functionality. Are you all keeping up with me? Ki are you all carried away?
Yes, ma'am.
Yes, ma'am, we are carried away.
No, ma'am, we are writing down.
Okay, fine.
No.
Yeah. So the first category is very basic, which helps in increasing and maintaining the drug solubility. All right. Second is to maintain chemical stability and physical stability. Okay. Fourth one is to maintain sterility. You can look at it as to maintain microbial stability. And the fifth one is miscellaneous. And this, do not take miscellaneous lightly, okay? Miscellaneous, maybe there are like treasures. And you know, ये जो तुम लोग books में पढ़ रहे हो excipients and reality में भी जो excipients use होते हैं, you all will be mesmerized by the way excipients are being used. And you know, as pharmacists, you all have lot of job opportunities, lot of role in excipient companies also. Do not restrict yourself only to the mainstream pharma companies or the mainstream non-core companies like IQVIA and all of that. Excipient company, maybe you have a major role in the R&D also. You have major role in what do you say, the marketing also. Techno commercial roles sune hai? You all understand what techno commercial roles are? Question mark, I'm asking you all.
No, ma'am.
Techno-commercial roles basically are wherein you need your strategic planning also, wherein you need marketing skills also, but you need a technical background also. And those roles are very good. If you are, suppose, an M.Pharm, and if you have very good communication skills and all, techno-commercial roles, very good, you make a lot of money. And you won't slog in R&D if you all don't want to work in lab. So these are some things you all need to look at. Okay. So anyways, coming back to the point. So which are the different categories? Okay. So now here, so the first branch of classification was on functionality. Now we will look at them categorically. कि इसमें कौन सी categories आती है. So increasing and maintaining drug solubility में you will have co-solvents, हमने 1000 बार पढ़ लिया है. Buffers, complexing agents, non-ionic surfactants in general. Okay. Chemical stability में you will have antioxidant, chelating agents, inert gases. All right. Physical stability में भी तुम्हारा surfactants and bovine serum albumin आएगा. There's a very interesting thing about BSA. Okay, this is called as BSA. Here, obviously, you will have antimicrobials. And miscellaneous में you have your cryoprotectants. As the name suggests, cryo or lyoprotectant will be used in lyophilization process. Okay. Lyophilization में क्यों use होता है? Is that suppose you are freezing your nanoparticles, suppose you are freezing your liposomes or even your peptides and proteins, उसको भी जब तुम freeze करते हो, you know, in the process of freezing, ice जब बनता है, it will be in form of crystal, right? It will form crystalline structure जैसा. Agree? So these crystals at times can get very sharp, and because of their sharp nature, they can rupture your liposomes, they can rupture the proteins and all of that. जिससे in the frozen state तो पता नहीं चलेगा, but वो rupture हो जाएगा, it will damage your proteins, it will damage your liposomes. And when you will reconstitute it, it will be degraded in a way. Okay? ये physical incompatibility हो गया, ये physical instability हो गया. So to protect it from these things, you use cryoprotectants and lyoprotectants. And even the type of cryoprotectant and the amount needs to be optimized. All right. Tonicity modifiers, obviously, to maintain isotonicity. All right. And the last thing we are going to look at are the examples. All right. So buffers में, you know, co-solvents के examples हमने last slide में पढ़ लिए. Buffers में, these are the commonly used buffers. These are the complexing agents. Caffeine is also used as a very good complexing agent. These are the surfactants. All right. So this is, yeah, these are pretty straightforward. Antioxidants are also divided into the ones which are used for aqueous preparation and which are used for non-aqueous preparation. Inert gas. Does anyone know how will inert gas help? Anyone?
To prevent oxidation.
Correct. Correct. So one of the chemical instability is oxidation, right? So when you are filling up your vial, okay, you're filling up the vial, obviously there'll be atmospheric oxygen, atmospheric gas, उसमें सब कुछ आ जाएगा. So oxygen भी जाएगा, and that oxygen can start oxidation and will start degrading your product. So to prevent that, what happens is that ये vial लिया, उसको half stopper किया, ये part खुला है. Okay, यहाँ तुम्हारा solution है. And here you will put a needle. And you will purge nitrogen. So purge or flush. नाइट्रोजन. ओके, so इससे क्या होगा, all the oxygen, because you're doing it forcefully, you're doing it externally, all the oxygen in the head space. I hope you'll understand what head space is. This is the head space जहाँ पे खाली
Yes,
हवा होती है. Okay, so all your oxygen in the head space will be removed and replaced by the nitrogen, and because nitrogen is inert, it won't lead to oxygen. And then when you're characterizing these vials, when you're testing these vials, you will also test for its oxygen content. So oxygen content जो acceptable है, it's not a fixed value. The level of oxygen जहाँ पे तुम्हारा product का degradation नहीं होगा, that will be acceptable for you. So at that level, तुम वो level एक set कर दोगे, and हमेशा वो nitrogen purging का rate, amount and all of that you will fix, and that you will use for all your products. Okay, so R&D में ये सब development चलता है. All right, so this is how inert gases will help. Yeah, one more thing I had to tell you all. And there's one instrument called as oxy-baby, which is used to find the oxygen content. So it's like imagine the device which is used to measure the in the drivers if they've had alcohol or not, right? I forgot what is it called. Okay, you breathe into it and then they tell you how much alcohol, you detect
Yes,
that, right? So it somewhat looks like that, just the thing it has a needle. So what you do is you put that device, it's somewhat like a mobile phone only, and say this is the needle here. Okay, this is the needle here. So in your vial, through the stopper, already stoppered है तुम्हारा vial, तुमने ऐसे prick किया उसको, and then you press the button, and then तुम्हारे device पे reading आ जाएगा कि कितना oxygen content. That is how you will detect. Always remember, this can be like an applicative thing in your MCQ, or just you need to understand this. Okay, whenever you are trying to get rid of something, you need to test it. In this case, you are trying to get rid of oxygen. So for that product, testing oxygen will become very important. And for that, you do even GC vagara you can do. So relatively, when you are trying to get rid of water, after lyophilization, what test will become important for your product besides other tests, which will be the additional test needed? समझा तुम लोगों को?
Moisture content.
Correct. Very good. Moisture content or water content. Okay, water content analysis will become very important. And which is the analytical technique which is used to determine water or moisture?
a Carl-Fisher titration.
Absolute. Beautiful. Who's this?
Chaitanya Carl.
I'm really impressed by Suraj, Yogesh, and Chaitanya. Okay, these are like, I don't know. Usually, girls are the ones who are answering all the time, but in this class, you three are, I'm really good. Please keep up this pace and this motivation. All right. So, for a lyophilized product, Carl Fisher testing or KF titration becomes very important. All right, to test the moisture content. I hope you all try to link these things up. When you are maintaining isotonicity, which is the test which will become very important for that aspect? Isotonicity के लिए कौन सा टेस्ट इंपॉर्टेंट हो जाएगा? अभी तारीफ की and you three kept quiet. What about others? Malika, Chaitanya नहीं, Chinmay, Andrea. Osmolarity test. Osmolarity test.
Osmolarity.
तो osmometer तुम वहां पे यूज करोगे. All right, so please try linking up analysis and chemistry with Sutix. It will get easy for you. दूसरी एक स्टोरी सुननी है फॉर अनदर एक्सिपिएंट के बोर हो गए?
Yes.
Yes,
सुनिए.
ma'am.
Yes, ma'am.
Okay. So another interesting thing about bovine serum albumin, and I don't know how well is it explained in your textbook. All these things are like, I'm speaking out of experience and my learning as well. All right. Bovine serum albumin का use physical stability में कहाँ आता है? It is very important when you're making biologics. Okay, biologics will include formulations like your vaccine, your peptides, your proteins, and all of those. उसको हम large molecules बोलते हैं. हाँ, biotechnological. Yes, उसको हम large molecules बोलते हैं. तुम्हारी जो सारे drugs and all है, they are small molecules. These are large molecules. Your peptides, proteins, vaccines, and all of that. So what happens is that all of these, your peptides and all of these, by and large, they are given parenterally. There are very few which are given orally. I'm not sure kitne hai, but I think there are very, very, very few which are given orally. Mostly they are given parenterally. Parenterally भी वो either lyo form में होता है या फिर there are some products जिसका lyo बनाना is not possible. So वो liquid form में होता है. So when it's in its liquid form, okay, so yeah, when it is in its liquid form, ये तुम्हारा glass vial है, ये solution में तुम्हारी ये peptides है, right? There's something like, then liquid form, they have some affinity to this glass. Okay, ये glass से उसका कुछ affinity होता है, ये proteins का, उनका nature वैसा होता है. So they will start agglomerating or they will start adhering to the glass wall. And proteins are so sensitive, these biologics are so sensitive कि मतलब even if you shake them, मतलब once they've adhered, once they start agglomerating, it's a way of them denaturing. मतलब they are breaking down, they are becoming physically unstable. All in all, they are losing their therapeutic activity. तो मतलब ये जो भी अधेर हो जाता है ग्लास को या फिर जो भी एग्लोमरेट होने जाता है, दे आर यूजलेस. तो उसका थेरेप्यूटिक एक्टिविटी पूरा डाउन. सो दिस इज नॉट गुड, राइट? सो व्हाट टू कॉम्बैट दिस, क्या बनाया है साइंटिस्ट ने? दे हैव मेड BSA कोटेड ग्लास वायल. BSA माने बोवाइन सीरम एल्ब्यूमिन. तो ये ग्लास वायल्स को उन लोग ने पहले ही बोवाइन सीरम से कोट कर दिया है. Obviously these glass vials will be costlier than the normal one. So they coat it with BSA, and this coating helps in preventing the adherence of these peptides and proteins to the glass vial. All right. Also, presence of BSA in the solution will help in preventing their agglomeration. So that is how BSA will help in improving the physical stability, especially for biologics. Understood, everyone? Hello.
Yes,
Any doubts?
Yes, ma'am. Ma'am,
Yes.
ma'am, so what will be the exact role of coating that inner wall of bovine serum albumin? Means that what we say intramolecular interaction won't take place in such case.
हाँ, मतलब जो ग्लास और प्रोटीन का, that's basically due to charge. Okay, the constituents of glass. Okay, glass also has some charge. I forgot if it's negative or positive. Okay, find it out. तुमने माइक्रोबायोलॉजी में पढ़ा रहेगा, gram staining and all that करते वक्त पढ़ा रहेगा. Glass also has some charge. Proteins also have some charge. So there's an interaction due to that. So BSA is going to prevent that.
Okay, ma'am.
Okay. So this is going to be an assignment question. Role of inert gases, role of BSA, role of lyoprotectant. And I want you all to cover up the points which I've explained you all. And something more also, if you all get it, textbook का छापा हुआ चीज नहीं चाहिए. मुझे पता चल जाएगा textbook का copy paste कैसा है. All right. This will tell me how much have you all understood. Explain it in short, that's fine. Got it?
Yes.
Yes, ma'am.
So yeah, these are the surfactants. And these are the few examples of cryoprotectants. Sucrose, trehalose. There are certain sugars. So these are the sugars only basically, which are being used. Carbohydrates be used हो रहे हैं आजकल as cryoprotectants. Amino acids like glycines and even lysine is being used. All right. The amino acid which is widely used for biologic products, yehi sab to maintain stability and pH and all of that is histidine. Huh? You all can make a note of that. Histidine. So let us just go through some of the preservatives very quick. These all are like easy slides, all right? So preservatives are something which will help you prevent the microbial contamination. Now I think you all are very much clear. तुम लोग को नींद में भी पूछ लिया. Which are the parenteral situations where you will not need a preservative? I think you all can enlist those major 5 points, right? Question mark, right?
Yes, ma'am.
Thank
Yes,
you.
ma'am.
Yes,
Thank you.
ma'am.
Yes, ma'am.
Okay. So, okay, so they are used in multi-dose containers and small volumes and, okay, to prevent microbial growth. So these are some microbials which are typically used in these concentrations. Remembering concentrations isn't that important. What you'll have to remember here is that chlorocresol these days is not being used. Phenyl mercuric thiamarosal, these again are very toxic, not being used these days. You all can find the composition. Mercuric में definitely mercury है, thiamarosal में भी mercury है, sulfur है. Benzyl alcohol is also benzyl alcohol is used. It's yeah, benzyl alcohol used in less concentration, not used for pediatric formulations. Benzoic acid is also used. Okay. Parabens these days are not being used because parabens are found to be carcinogenic. Your textbooks still have them, but parabens are not preferred. However, there are some cases where it can be used. Yeah. Besides, benzyl alcohol is also used in your ophthalmic preparations. So yeah, here again you have this. कहाँ पे कौन सा यूज किया जाता है? Oleogenous preparations माने कि your oil preparation. वहां पे you have your resorcinol, phenyl mercuric used as your bactericidal and all. ठीक है? So this is and benzoic acid is also used for ophthalmic preparation. So this is about your preservatives. ये table तुम लोग को this is a little important. Okay. Antioxidants, they prevent oxidation, obviously. So what happens is, does anyone know how does an antioxidant play a role?
They itself get oxidized.
Correct. वो
And they prevent the formation of the reactive oxygen species, ROS.
the correct term here is ROS, reactive oxygen species, and O2 radical, oxygen radical.
Oxygen
So
radical.
these are basic. ROS is basically oxygen radical. All right. You all know what radical is, right? One free electron, if I'm not wrong.
Yes.
Yeah. So उसका formation भी prevent करता है. And who answered it gets oxidized itself? So yeah, Suraj. Yeah. So both are correct answer. वो खुद sacrifice हो जाएगा. It's the mummy of the mother of the excipient. खुद she will tolerate. All right. So that is how it prevents oxidation, reduces the oxidative stress, and obviously imparts physical, chemical stability to the formulation. Okay. And how? Why does it get oxidized by itself? Because they have a lower oxidation potential than the drug. If you all go back to your 12th standard chemistry into the nurse potential, okay, electrochemistry, redox, both are the topics if you all go back to nurse potential and all, you will have a list. Okay. So the ones which have a lower oxidation potential, they will get oxidized first. So that is what antioxidants basically are. And these are the antioxidants which are typically used. One more I will add to the list because that I'm using in my lab, which is propyl gallate. Okay, so even these are the antioxidants used. नाइट्रोजन गैस को मैंने इधर इसलिए प्लेस किया, इट्स नॉट एन एंटीऑक्सीडेंट, it's basically an inert gas which will prevent oxidation. Okay, so it's an indirect antioxidant, you can say, and I hope you all are clear with it. Okay, okay, so these alpha tocopherol, this is used in oily preparations. BHA BHT is also used in oily preparation. Ascorbic is used in aqueous. All these are used in aqueous. Propyl gallate is also used in oily. I'm not sure about aqueous and all. All right, so these are the antioxidants. I want to show one diagram to you all. Acha, wo idhar hai. Next slide. You all can see the slides, right? You all can see the slides, question mark.
Yes, ma'am.
Okay. Okay. So antioxidants की बहन is chelating agent. Okay. ये मौसी है excipients की. So what will even chelating agents do? They जो भी metal ions in case they are present in a formulation. Okay. जो भी metal ions present है in the solution में, they are not favorable for your formulation. Okay. Even metal ions lead to oxidation. Okay. Even metal ions lead to oxidation. So what will chelating agents do? Instead of the metal ions reacting with your drug, okay, here in this case retinol, reacting with your drug and degrading it, chelating agents खुद metal ions के साथ chelate form कर देगा, okay, या complex form कर देगा, and it won't let them react with your drug. So they basically steal their electrons. All right. So this cartoon is trying to depict that. So yeah, these are the major ones: copper, iron, manganese, which are present usually, prevent oxidation and thus improves chemical stability. Okay. Question: what is the type of bond which is formed by the chelating agent?
Coordination bond.
Correct.
Coordination.
Perfect. Perfect, Chinmay. And others. All right. Coordination bond is the bond which is formed by the chelating agents with the metal ions. All right. And these EDTA is commonly used one. Citric acid is a commonly used one. All right. So this is how it's going to improve the stability. Right? Yeah. Now you have your buffers, pH stable. Parenterals in general should be 3 to 9. Now it will differ for ophthalmic, IV, differ for intra-articular and all of that. All right. SCIM का भी pH value differ करेगा. Tissue to tissue pH value differ करेगा. ठीक है? So these are the different pH buffering agents and all being used. Okay. Then these are the suspending and the wetting agents done. Okay. This is in order to maintain some amount of viscosity. This is for basically for your suspension or your nanoparticulate novel drug delivery systems के लिए, nanoparticle liposomes के लिए. Okay. So हमने suspensions पढ़ा है. So we know what is the role of suspending agents. It will impart viscosity and prevent in agglomeration and prevent sedimentation and all of that. Povidone is widely used. CMCs are not that widely used. Yeah, now surfactants surfactants, one surfactant can play multiple roles. Surfactants can help you increase solubility. It will help help you in maintaining viscosity and all that also. So surfactant का multiple roles हो जाएगा. So this was about excipients. Any doubt?
Ma'am,
Yeah.
what is the point of histidine? One
Sorry?
point you mentioned. The histidine as an amino acid is widely used in.
It's kindly used during some buffering action for biology.
Okay.
Okay, that was the only thing.
Yes. Yes.
ठीक है. So इसके basis पे let us solve some MCQ. Any doubts in this excipient?
Ma'am, at a one time,
Yeah. हाँ.
A particular excipient can give a role of two things. For example, that tocopherol. It is
Yeah.
also an antioxidant as well as used as a surfactant. So,
Tocopherol is not used as a surfactant.
ma'am, for antioxidant.
Tocopherol is used as an antioxidant. Agree. What are you trying to ask? Come again.
मैम मतलब एक एक्सिपिएंट का एक से ज्यादा रोल होता है तो चल जाएगा ना, मतलब उतना ही प्रोसेस थोड़ा कम कॉम्प्लेक्स हो जाएगा, राइट?
प्रोसेस
ज्यादा
कम कॉम्प्लेक्स नहीं, मतलब इट्स लाइक एक तीर से दो निशान. उतने कम एक्सिपिएंट्स यू विल हैव टू यूज बिकॉज एक ही एक्सिपिएंट से तुम्हारे दो काम हो रहे हैं, देन दैट्स बेटर ओनली ना.
ओके.
Okay. Now, tocopherol is used as a supplement also, vitamin E. So in that formulation, you don't have to add additional antioxidant. Maybe you can just purge the system a little with nitrogen and then you can have it. There are, many drugs and drugs which have they have their own antioxidant property also. तो उन केस में तुम्हें एंटीऑक्सीडेंट शायद यूज ना करना पड़े या करना भी पड़े तो बहुत कम यूज करना पड़े. Got it?
Yes,
Anyone? Anybody else? Any other doubt?
No, ma'am.
Okay, so let us solve MCQ based on this part. All right. Do you all think I'm going slow or anything?
no ma'am, this space is perfect.
No, ma'am. Perfect.
Proper है ना?
Yes, ma'am.
Okay. If you all feel I'm going fast or slow now, please let me know. So we stopped at question number 12, right? Are your MCQ books open?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
You all can see the slides, no?
No, ma'am.
No. Okay. अभी दिख रहा है?
Yes.
Okay, so we did question 12. Now we are going to question 13. All right. Which of these is not a parenteral component? Sorry. Question 13, okay? आंख खोल के दिमाग खोल के आंसर करो. The correct answer here is E. All of these are components. Container you all will agree, API you all will agree, vehicle you all will agree. Just because मैंने emulsifier list नहीं किया, just a minute. Just because मैंने emulsifiers list नहीं किया doesn't mean it's not used. Emulsions दिया जाता है कि नहीं parenteral में?
Yes, ma'am.
Yes, जाता है.
Then emulsifier use होगा कि नहीं होगा?
होगा, ma'am.
right? So please think in that way. Okay, just because I'm not enlisted it, that's why this is an MCQ here. Okay, be alert while answering. Okay, ratta mat maro. All right, moving to question number 14. Select the correct pair. So select all correct options. Okay, so there can be more than one answer. Come on. The answer is A and B. लो मॉलिक्यूलर वेट का तुम रिवर्स ऑस्मोसिस कैसे करोगे? तो उसका ही वो ही फिल्टर आउट हो जाएगा ना. रिवर्स ऑस्मोसिस में भी क्या होता है? देयर इज मूवमेंट ऑफ वाटर एंड उसके साथ साथ तुम्हारा अगर छोटा साइज है पायरोजेंस का तो वो भी मूव हो जाएगा. All right. Even though I said reverse osmosis is a thing used for uh, uh, clearing, but it's not because of its low molecular weight. All right. So these two are correct pairs here. Question 15. Make it quick. Please write the question number also. Which is true for the vehicle? There can be more than two answers. So water is preferred, but only WFI grade, correct? Okay. Vegetable oil is not preferred because they turn rancid. Correct.
But they are preferred. No?
They are, you will avoid it, right? Right? Oils cannot be used in, okay? So what is true? This is true. You keep a check on the water miscible solvents, but it can be toxic, right? This is when I taught you all liquid preparations. तभी हमने co-solvency की बात की थी. मैंने तभी भी बोला था कि these co-solvents are very good, but you have to use them very rationally because they can be toxic. All right. And तभी मैंने IIG के बारे में बोला था, inactive ingredient guidelines, and it should be GRAS, generally regarded as safe, right? And obviously oils cannot be used in some cases. So these three are the correct answers here. Question 16. So here actually all are correct. Complexing of API, right? Tumhara HP beta CD, that is your cyclodextrin. Water miscible solvents, co-solvency, solubilizing agents, obviously. Even buffers, if you have pH dependent solubility of the drug. So तुम ऐसा buffer, ऐसा pH maintain करोगे, which will help in improving the solubility. Okay. So all of these are correct. 17. Question number 17. Are you all there?
Yes,
Okay,
ma'am.
abhi idhar
Yes.
only one answer. Only one answer. Yes. Correct. The correct answer is B. Parenteral API must be non-pyrogenic. तुम किसी में भी नहीं बना सकते हो. It has to be specially dedicated for parenteral preparation and may have some chemical impurities, not at all. All right. This I just told you, all right? Yeah, histidine is the correct. Surfactant used in parenteral. This is for your understanding. हाँ ये वाला and जो मैं multiple वाले देती हूँ ना, they won't be. This is for your understanding so that you all can have some applicative response to the question. Question 19. Yes. D. Based on suitability, you will decide कौन सा use किया जाता है. Okay. So based on suitability, you will decide what surfactant you want to use. Based on suitability and based on the use, तुम्हें क्या application के लिए use करना है. Because surfactant solubilizer का भी काम करेगा, suspending agent का भी काम करेगा, emulsifier का भी काम करेगा. Okay. तो तुम्हारा application क्या है, तुम accordingly use करो. And aqueous के लिए है कि non aqueous के लिए है. ठीक है. Okay, we will take. You'll want to solve it now or should we do it in the next class from 20?
Next class.
Yeah. ना overdose हो जाए. ठीक है. So did you all understand today's class, everyone?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, I'm assuming it no.
Yes.
Okay, fine. Anyways, so you all need to you all will get the study material for this second lecture by tomorrow. If you all don't get it by tomorrow morning, contact your admin. I will upload it. And by tomorrow, I will also upload the assignment questions by today or tomorrow. So solve it accordingly. And as I told, upload in either one image or one PDF. Okay? And yeah, that's it. Any doubt?
No, ma'am.
All right. Bye-bye. Good night.
Thank you, ma'am.
Bye, ma'am.
Thank you, ma'am.
Bye, ma'am.
Thank you, ma'am.
Thank you.